Good afternoon, everyone. I think in the interest of time, we're going to get started with our webinar today. Welcome. My name is Vicki Ellingrod. I am the Senior Associate Dean in the College of Pharmacy and also the Education Work Group Lead for Precision Health. Before we get on to our two wonderful speakers today, I wanted to say a few words about precision health. Started nearly 3 years ago, precision health is really focused on building infrastructure and providing tools that enable interdisciplinary research across campus. And you're going to be hearing some great examples of this research very shortly. If you would like some other examples of some of the resources that Precision Health has to offer you as a researcher, you can head on over to the webpage where you can find many tutorials that have been produced to aid investigators and their students regarding how to use the robust analytics platform to help researchers from all disciplines access and use precision health data. Precision Health has also been encouraging research with funding and educational events on a regular basis. And in fact, currently Precision Health has already distributed nearly $6.5 million in research funds here on campus. In addition, the Education Task Force has developed a Precision Health Certificate Program, which is the first in the nation focusing on precision health. It is open to all graduate and professional students across campus, and we will soon be enrolling students into this program for the fall. So keep an eye out for those announcements as well, or head on over to our website to learn more about this really innovative and interesting program. Last but not least, Precision Health has been working to implement the findings from some of their supported research projects, as well as from some investigators here on campus, into real-life healthcare settings. This is being done in an effort to impact patient safety and improve overall patient outcomes. If you're interested in becoming a Precision Health member, we invite you to join using the link that you see here on the screen or going to our Precision Health website and following the instructions that are on the website. We have nearly 200 members now, and they are able to use all of our resources, but more importantly, they get direct communications from Precision Health to help publicize their work, as well as learn about all the benefits of being a member of Precision Health. So again, please use the link that's here on the slide, or go to the webpage to learn more about how to become a member of Precision Health. So today we have two really wonderful speakers. We are going to, each speaker is going to be allowed about 20 minutes to give their presentation, followed by some time for question and answers at the end. If you have questions for each of our speakers, please submit them either through the chat box or through the Q&A function on the bottom of your Zoom screen. This session is best viewed if you go into the speaker view. And if you have questions about how to do that, it's on the upper right-hand corner of your Zoom screen where you see the view and click speaker view. Again, if you have any questions for the presenters today, please feel free to put those in the chat box, and then I will be reading the questions to our speakers as part of their Q&A session. So today we're excited to host two of our early career researchers, Alyse Krausz and Arun Subramaniyan. Both have received funding from the Precision Health Initiative through the Scholars Award Program. Our first speaker today is going to be Arun. He is a 2018 Precision Health Scholar. He's a fifth-year PhD student in the College of Engineering who is being advised by Professor Das. His dissertation research focuses on developing efficient algorithms and customized computing systems for precision health. His work has been recognized by Rackham's International Student Fellowship, as well as the IEEE Micro Top Picks Award, which is an award given annually to significant research papers in computer architecture. And with that, I am going to ask Arun to share his slides, and I am going to sit back and enjoy your presentation. Thank you.
Thank you for the introduction, Vicki. Is everyone able to see my slides?
Yes, we are.
Okay, thank you. So I'm really excited to present some of our work over the last 1 or 2 years, in which we have been trying to design much more efficient algorithms, as well as systems that can efficiently process data from specific to precision health and in general sequencing data. So let's get started. So sequencing is one of the key ingredients to enable the precision health revolution. And over the last decade or so, we have seen an exponential growth in the amount of sequencing data, starting from the Solexa genome analyzer in 2005, all the way to PacBio and MinION sequencing developed in 2015, and single cell sequencing that is very common today. So we have seen that sequencing data has been growing roughly at the rate of doubling twice every 12 months. And Illumina projects that it would also increase even faster in the future. So along with this increase in sequencing data, there has also been a commensurate decrease in the cost to sequence a human genome. So over the last decade or so, there has been more than 1000x decrease in the cost to sequence one single human genome. However, data analysis that is trying to make sense or figuring out the differences between a sequenced individual and a reference genome has not been able to keep pace. So in this slide below, you can see that sequencing data from sequencing machines is produced roughly at the rate of around 350 genomes per week. But computing systems that are analyzing the sequencing data are able to only keep up at the rate of 12 genomes per week for a given computing server. So we looked at this bottleneck and are trying to figure out how we can improve the efficiency of computing systems so that they can keep up with sequencing machine throughput and the volume of sequencing data that is exponentially increasing. So in over the last 20 years or even before that, Moore's law was coming to the rescue. So we did not have to wait to develop better algorithms or try to develop better systems, because in the next 2 years, your system would be upgraded and it would already be automatically twice as fast. But unfortunately, the Moore's law, which is the observation that transistors become twice as fast every 18 months, or the number of transistors on your chip doubles every 18 months, is no longer true. So what is happening today is that the performance of computing systems is roughly increasing at the rate of roughly doubling only once every 20 years, as opposed to 18 months. So therefore, to keep up with the sequencing data that is doubling every 12 months, we would need a significant increase in the efficiency of computing systems today. Also, sequencing data is of different kinds. It's highly diverse, and sequencing pipelines are also significantly diverse. So now the volume of sequencing data produces roughly of the order of few terabases per day, which is 1000x increase from what was nearly a decade ago. We also have portable sequencers such as those from Oxford Nanopore, which are producing longer reads, but because of their error profile, which is slightly higher than Illumina-based sequencing, the algorithms that are used to process long read data are also need to be error resistant or error tolerant, so that we can still extract insights from this new kind of sequencing data. So given all of these trends, both in terms of the volume as well as the diversity in sequencing data, systems for computing on this data and trying to extract insights have three main requirements. So they have to be much more efficient, as I've shown in the previous slides. So we need to come up with ways in which we can process, say, several genomes in parallel across several different machines, or try to make each one of those machines much more efficient, So that's one approach. And this way we can improve both the performance as well as the energy efficiency. Another approach is to also try to reduce the storage cost because the cost to store this large amount of genomic data is increasing exponentially. We need to come up with better compression algorithms so that we can achieve much better storage efficiency as well. Also, with genomic data and data sharing between several institutes and government centers, computing on this data must not reveal any identity related information from each of the shareholders or stakeholders. So we need to come up with algorithms that can efficiently operate on both the encrypted data as well as uh, normal data as well. And another challenge is that we are now having sequencing devices which are much smaller form factors, such as those from Oxford Nanopore. So we need to also integrate our computing systems in such a small form factor, like inside a mobile phone or inside a USB dongle. So putting computing systems in such a small form factor makes us forces us to think about how we can redesign our systems to be much more energy efficient than what we have today. So in our work over the last year and a half, we have been mainly looking at how we can improve the compute efficiency of systems for analyzing sequencing data. So let's look at how we did that. So the silicon technology that we have today, like our conventional multi-core CPUs, uh, they can run web browsers, they can process desktop applications, you can run media players on them, but they have poor performance and energy efficiency. On the other hand, GPUs or gaming systems or custom hardware for compression provides much better performance per watt of energy consumed, but they are comparatively less programmable. At the other end of the spectrum, you have custom hardware that perform a given task very efficiently, such as video processing or embedded system-based pattern recognition modules. So, but as you can see, there's a wide spectrum of available hardware, and as you go down, they become much more performance and energy efficient, but at the same time, less flexible. So in our work, we have been trying to optimize both existing software that run on conventional CPUs by redesigning existing data structures and algorithms, as well as trying to map these algorithms to custom hardware so that we can take advantage of their increased performance and energy efficiency. Specialized hardware has been already leveraged for machine learning. So Google has been using the tensor processing unit hardware to accelerate Google search. And Microsoft has been using Catapult-based FPGAs, which is used to accelerate Bing search in its data centers. Genomics is also not far behind. There have been companies such as Parabricks from Ann Arbor itself, and Illumina, which recently acquired the Dragon-based platform developed by Edico Genome to accelerate sequence analysis. So there is an increasing trend to also leverage custom hardware to accelerate genomic analysis. And this is inspired by this trend. We also try to explore how we can leverage custom hardware to accelerate some of the steps in sequencing analysis. So before we dive into what we did, let's look at a conventional secondary analysis pipeline. So you have a sample taken from a patient, let's say, and after sequencing, you get billions of short reads, say 100 characters long. These reads now go through a series of computational steps. The tools that are most common for these steps are shown below as well. And after a series of processing steps known as read alignment, sorting and duplicate marking, and variant calling, we finally have data presented in a form that is now amenable to downstream analysis as well as diagnosis. So we looked at this pipeline and we found that on a single multi-core processor, which has 32 threads, let's say, all processing one genome takes about 13 hours. And the breakdown into different steps is shown below as well. And you can see that almost all of the different steps are taking roughly the same amount of time. So we will look at how we can reduce the time spent to process one genome. So first, let's look at read alignment. So read alignment is one of the major bottlenecks, as we have seen in the previous slide. And it consists of two main steps. One is called seeding and the other is seed extension. And together in software like BWMM, these take 200 CPU hours for a single 50x coverage human genome. So how does seeding work? So seeding tries to find the location of short matching strings in your read in the reference genome. So this allows you to start finding the optimum location for the read by taking smaller substrings from the read called seeds and trying to match their locations in the reference genome. So the seeding step is typically performed using a data structure called the FM index, and the algorithms using the FM index conventionally are memory bandwidth bottlenecked. This is why they're time consuming and they take a significant amount of time. But they're highly compressed, and because so that they can fit on even small laptops and you can process your sequencing data. What we notice is that if we can trade off the size of the index, but reduce the memory bandwidth consumed, we can improve the performance of the algorithm by several fold. So our first observation was that we would trade off memory capacity, but to reduce memory bandwidth so that we can improve the overall performance of the algorithm. So we implemented this new data structure and algorithm in software BWMM2, and we found that we got identical output as the baseline software. Uh, we went a step further and worked with the developers of BWMM2 and tried to integrate our code into the original repository. And for this, we had to leverage some of the resources that we got through the Precision Health Award to run extensive experiments on the cloud so that we could validate all our results and ensure that we can meet the quality standard of analysis software such as BWMM2. So the next step after seeding is seed extension. And this is an approximate string matching step, which tries to find the best location for a given read, given all its constituent seeds. So how does approximate string matching work? It tries to find the best possible alignment between a pair of strings, assuming a certain number of edits, k. The conventional algorithm has complexity, both time and space, of the order of k times n. And we came up with another algorithm that can process these sequence pairs in just k square complexity. And this work was published in ISCA 2018. It supports several of the features that are needed in read alignment tools, such as affine gap scoring and traceback. And to demonstrate proof of concept, we also fabricated this chip in 55 nanometers. Processing using this chip is 63 times faster than using a multi-core CPU. Next, to look at how we accelerated variant calling, we found that it's dominated by floating point computation, which is expensive to implement both in conventional CPUs as well as custom hardware. So we try to look at whether we can replace some of the floating point computation in this workload by fixed point low precision computation. And by doing that, we were able to significantly reduce the number of cells that needed to be computed in precise floating point representation by 43x. And as a result, our accelerator for this algorithm, which is the pairwise hidden Markov model, or pair HMM in short, was able to achieve 8.3 times higher throughput than another ASIC that computed the full precision for the same area budget. So putting all these accelerated implementations together, we found that we were able to reduce the time to sequence and analyze analyze a given genome by in less than 2.5 hours, which is a 5.5x improvement over the baseline CPU. Moving on to some of the work that we're currently doing, we noticed that several of the researchers in the computer architecture and computer engineering space are unaware of the characteristics of genomics workloads, which are increasingly becoming important both in academia and industry. So we collaborated with Intel to develop Genomics Bench, which is a set of computationally intensive algorithms drawn from several software tools. And this is open source as well. And we encourage you to try it out if you're interested in looking at the computational challenges for emerging genomic workloads. Apart from this, another interesting project that we're working on is in collaboration with Dr. Lona Modi and Dr. Betsy Foxman from the medical school. And we are trying to figure out if we can analyze host pathogen interactions, specifically the transcriptomic interactions, and try to figure out if we can stratify these patients into colonized, mildly infected, and patients that are likely to get a severe disease. Our goal is to determine a set of biomarkers, both from the host and pathogen side, that can be used to distinguish colonization from infection in these elderly patients, and also predict in advance the risk of infection for different patients. I'd like to thank Anissa and Kate for helping us with the data direct and familiarizing us with the tools present there, and that was really helpful for this project. So this work would not have been possible without our excellent collaborators as well as the graduate students in our group. So I'd like to thank both my advisor, Professor Das, as well as Professor Satish and David, as well as collaborators from medical school and Intel. So with that, I'll stop and happy to take any questions that you may have.
Thank you very much for that presentation. Wonderful presentation. If anyone has any questions, please feel free to put them in the chat. I have more of a comment than I do a question, in that it's as someone who grew up with IEEE magazine in my house, because my father was an engineer. I think it's really amazing that you're talking about being able to do these sorts of analyses on something the size of a cell phone.
Right.
And I think it's the finding efficiencies in not only the human resources, but also the kind of the compute environment, and how do we do this to advance science is really exciting. That we're at that point now. Could you tell us a little bit more about the pneumonia project that you're doing? Is that looking in the electronic health record as part of it? And then what are some of the other things that you're looking at as part of that project?
Right. Yeah. So for that project, we are trying to see how we can integrate different sources of data, both electronic health records as well as sequencing data, and try to develop predictors that can figure out the state of a patient. So we have been trying to leverage data direct for this, and trying to see whether we can develop proof of concept models using already available electronic health record data. And that's where we are right now. And once we get sequencing data as well, we will try to correlate with the EHR data as well as try and improve predictions only based on EHR.
So is the vision of that then kind of doing the using AI resources to kind of continually run in the background and identify patients at risk before they reach? Oh, cool. That's exactly very interesting.
Right.
I see we have a question in the chat. How does the reference equipment do the sequencing so fast? Any computing used there?
Okay, by reference equipment, yeah. So let me try to understand the question as well.
I think you can read it in the Q&A too if you want to open it up.
Right, let me do that. So
okay.
How does the reference equipment do the sequencing so fast? Any computation used there? Yeah, so I would encourage you to look at all the references which I put in the several slides, and once the recording is available online as well, you can take a look at the references. So all of the algorithms and the changes that we did for each of them, so that we can improve its performance on both CPUs and GPUs. Okay, the equipment you were showing in the beginning. Right. Okay. Yeah, I think. Yeah, I would encourage you to look at it, and I'm happy to also answer this question offline. So please reach out to me if you want any more information. So yeah, I guess at high level, we try to analyze the algorithm in depth and try to figure out opportunities where the hardware was not used efficiently, and try to redesign the algorithm so that we can improve its performance.
Are there other questions from our audience? Currently, I think we have about a minute or two left before we need to move on to our next speaker. Okay. Well, with that, I want to thank you once again for a wonderful presentation, and it's so great to see some of the, the primary resources provided by Precision Health, and how you're trying to improve on those, and then getting to the point where developing algorithms that could be potentially implemented within the health system to improve patient care. So I think that that's a really great example of the type of projects that Precision Health is trying to support. So thank you very much.
Yeah, thank you.
And with that, we are going to move on to our next speaker today. So I would like to introduce Elise Krauss, who graduated from The Ohio State University in 2016 with a BS in Biomedical Engineering. She's currently finishing up her PhD in Biomedical Engineering in the lab of Dr. Mark Burns. And she plans to defend her dissertation in July, which, yikes, is really coming up.
Yes.
The focus of her research is on the development of point-of-care microfluidic technologies. And she was previously a microfluidics in biomedical sciences training program fellow. Again, similar, if you have questions, please feel free to put them in the chat or the Q&A. And with that, I am going to turn my video off and mute and take it away, Elise. Thank you.
Thank you, Vicki. So today I'm going to be presenting on my work in developing a variable height microfluidic device for multiplexed immunoassay analysis of traumatic brain injury biomarkers. So there we go. Traumatic brain injury is ubiquitous, meaning that it can happen to anyone at any time, and it can range in severity from something more mild, like a concussion, to something more severe that could lead to a coma. And these injuries are commonly caused by falls, car accidents, a sporting injury, or a blast injury, which has become the signature injury of the conflicts in Iraq and Afghanistan. And it's estimated that there are about 2.87 million cases in the US annually. And this number is almost certainly an underestimate because this just takes into account people who sought medical treatment after their injury. And many people who suffer a concussion might not know that they suffered a concussion, and then they never seek medical treatment. But if a patient does seek medical treatment, a physician will usually start out with a physical examination. And so they'll examine a patient's eye-opening response, their verbal response, their motor response, and then that patient will be assigned a score on the Glasgow Coma Scale. So on this scale, the lower the score, the more severe the injury. And as the name implies, this scale was originally used to determine if someone was in a coma or not, but now it's commonly used to assign injury severity in a traumatic brain injury, so mild, moderate, or severe. A physician might also order a CT scan or MRI to look for physical tissue damage in the brain, but for a milder injury like a concussion, tissue damage might not be visible on the scan because the scan just isn't sensitive enough to pick up a more diffuse injury. And finally, an up-and-coming method of traumatic brain injury assessment is blood biomarkers, specifically proteins. And these biomarkers reveal tissue damage at the cellular level. And these biomarkers are constantly being discovered and validated. And each one can provide different injury information. So for example, UCHL1 and NFL are released from neuron cell bodies after an injury. Tau is released from the axons. GFAP is released from astrocytes, which are a support cell for neurons in the brain. And then on the other side of the blood-brain barrier, there's going to be an inflammatory response going on because of this injury. And so that can be monitored using IL-6 and IL-8. And there are many biomarkers being validated, but there are currently 2 that are FDA authorized, and those are GFAP and UCHL1. So these 2 biomarkers can be used to determine whether or not someone needs a CT scan if they've suffered a concussion. But we hypothesize that these biomarkers could be used for more than ruling out the need for a CT. And to illustrate that, I'm showing you a graph of serum concentration of UCHL1 in yellow and GFAP in navy over time after an injury. So you can see that patients without a concussion, so a mild to moderate traumatic brain injury, these protein biomarker concentrations are relatively constant over time. But for someone with a mild to moderate traumatic brain injury, so a concussion, these biomarker concentrations peak at certain time points. And this time course of the concentration could potentially be used to determine injury severity by looking at the magnitude of this concentration peak, as well as the prognosis for a given patient based on how long it takes for these biomarker concentrations to return to baseline levels. And so this biomarker data is becoming clinically available. These biomarkers can currently be measured using a Quanterix analyzer that's meant to be used in a hospital laboratory, as well as a point-of-care device, the iStat Alinity. This was just FDA approved to measure GFAP and UCHL1 back in January. But the problem with both of these methods is that they can only be used in a hospital setting. And so we're developing a device that can be used to measure traumatic brain injury biomarkers in clinical and non-clinical settings. So for example, on the sidelines of a football game to determine whether or not a player can go back in. And so to enable this measurement in non-clinical settings, we need the device to have a small footprint, be able to measure these biomarkers quickly, measure multiple biomarkers at the same time, and be able to read out results on the device itself as well as transmit them. So for this presentation, I'm going to be focusing on number 1 and number 3. So this is what our microfluidic device looks like now. And you can see there's a band here, and that is a collection of 2.8 micrometer assay beads that have a sandwich immunoassay on them for GFAP. And the signal is a fluorescent quantum dot. So when we analyze the fluorescence of this band of beads, the intensity corresponds to the concentration of the protein biomarker of interest. So I'm going to break down each of these components of the system, starting with the assay itself. So we developed a CLISA for GFAP rather than an ELISA because we needed this fluorescent signal to be directly tethered to this bead. If we had used an enzyme, that fluorescent product is not directly tethered to the bead and it's free to diffuse away. And we didn't design the chip to have any barriers in it. And so that would decrease the sensitivity of the assay. So that's the assay. And so now let's talk about the chip to run the assay. We fabricate the chip using glass wafers, which is this glass disc here, and we pattern them so the glass is exposed where we want our devices to be. And then we slowly lower this glass into hydrofluoric acid. So different parts of this channel are exposed to the acid for different amounts of time. So that means we end up with a height gradient across that channel. And this is different from how people typically etch glass for microfluidic devices. Typically, people will just submerge this entire wafer into the hydrofluoric acid for a set amount of time, so they end up with the same height profile all the way across. And so after that etching process, we end up with 3 sides of the channel. So we need to close it off and add a 4th side. And so we do that by bonding the etched glass wafer to a silicon wafer that we've mechanically drilled holes into. And so this is what the completed wafer looks like. And one of these channels here is considered to be 1 chip. So we will be eventually dicing these out. So you end up with like a test strip looking device. And you can see that there are these bands here, but those are not beads. That's a diffraction pattern due to the varying height between the inlet and outlet. So if we see these diffraction patterns, that means that these bonded correctly. So if we zoom in on this channel here, we can see that the device is 3 millimeters wide and 2 centimeters long between the inlet and outlet. And I've lined this height graph up with the channel such that you can see that the band of 2.8 micrometer beads that are trapped here, they get trapped at the point where their diameter matches the height of the channel. So that's the assay and the device. So let's put them together so we can actually analyze this for protein. So I developed a standard curve for GFAP using 2.8 micrometer assay beads, and the fluorescent signal is from quantum dot 585, which is a yellow, represented by yellow here. And I got the standard curve by analyzing the fluorescence of the band of beads trapped in a channel. And this is done in spiked serum samples. I also developed a ELISA for IL-6 using 4.5 micrometer diameter assay beads and quantum dot 525 as the signal, represented by cyan. And then the third ELISA I developed is for IL-8, which is an inflammatory biomarker. And the assay bead diameter here is 1 micrometer, and the fluorescent signal is from quantum dot 655, and that's represented by magenta. So I now have 3 different assays for 3 traumatic brain injury biomarkers using 3 different sized assay beads. And so that really enables multiplexing because this device has many different heights along its length. So I can introduce a mixture of these assay beads and have them separate out into distinct bands as they flow through the channel. And so what I end up with essentially is what I like to call a traumatic brain injury barcode. So to illustrate this, I did a 2-plex of IL-6 and GFAP. So this band here is for IL-6, and this band here is for GFAP. So this is a representative fluorescent image taken from this band, and I'm overlaying the fluorescence that we expect from IL-6 with the fluorescence we expect from GFAP here. And the reason I'm doing that is because I want to see if any beads got trapped in this band. Because like I said, these bands are forming simultaneously. So these small beads have to go through the larger beads as that band of larger beads is forming. So potentially some of those smaller beads could become trapped in the band of larger beads. And so we can see 2 things in these images, some bead crossover as well as some autofluorescence. So if we zoom in on this image here, we can see that there are some yellow beads. And that's because some of those smaller 2.8 micrometer beads got trapped within the 4.5 micrometer beads. And if we look at this downstream band, we'll see that there's some cyan and there's yellow. And that cyan is not from 4.5 micrometer assay beads because they physically cannot make it that far down the channel. It's from autofluorescence because unfortunately the surface of these beads autofluoresces at 525 nanometers. So to quantify what I was showing you on the previous slide, this is the fluorescent intensity from that upstream band, the IL-6 detection band. And this is the signal we're expecting. And then this is the signal from some bead crossover. And if we look at the downstream band, the GFAP detection band, we can see that there's a large contribution from 525 nanometer emission. And this is the signal we're expecting here, this yellow bar. So if we look at this set of bars from the previous slide compared to a sample that had no GFAP in it, we can see that this signal in the 525 nanometer channel is conserved regardless of how much protein is present. And so we conclude that this is because of autofluorescence of the surface of the beads. And so back to that original bar graph I showed you, this is from bead crossover and this is from autofluorescence. But luckily that bead crossover contribution is minimal, and so we're able to multiplex these assays. And now, because I was successfully able to perform a 2-plex, I moved on to performing a 3-plex of all 3 assays for the protein biomarkers. And so in this case, IL-6 is the same, GFAP is the same, and I've added in the IL-8 assay with the 1 micrometer bead. And again, we see some bead crossover and autofluorescence. If I zoom in, you can see that a little better. There's bead crossover of the magenta and yellow beads in the upstream band, and in the middle band, there's crossover with the magenta beads. And then again, you see autofluorescence in the cyan channel, in the GFAP detection band, as well as in the IL-8 detection band. And again, this is just to quantify what I showed you on the previous slide. There's some bead crossover in the upstream bands and some autofluorescence in the downstream bands. So at this point, you might be wondering, I am physically separating out these different bead sizes, but then I'm also using 3 different colored quantum dots, which kind of seems maybe like overkill. But the reason I did that was because I wanted to easily visualize that bead crossover. So that's easy to do if I'm using different fluorescent signals for each assay. But I don't need to do that, and I'm not planning on doing that when this is implemented at the point of care. So to illustrate this, I used 1 quantum dot color to multiplex both IL-6. So the IL-6 signal is now yellow, and the GFAP signal is still yellow. And I was able to distinguish between different concentrations of these biomarkers. So this is the set of images I showed you on the previous slide, and each set of images is from a different channel. So this was a mixture of beads that separated out, as well as these other pairs. So you can see that I'm able to distinguish between different concentrations, but I would like to point out that we still see some bead crossover here. And at 0 picograms per milliliter of protein, we do start to see some quantum dot aggregates. And fortunately, these quantum dot aggregates and bead crossover does not impact the overall concentration measurement because of our image analysis procedure. My image analysis procedure ignores beads that are smaller than what we would expect to see and objects that are larger than what we would expect to see. So to wrap up kind of what I've talked about, blood biomarkers are a developing area of TBI assessment. CLISAs enable rapid and sensitive biomarker quantification. And those CLISAs can be multiplexed in our variable height device because that variable glass etching procedure produces a device with intrinsic multiplexing capabilities. So we have this device, but so what? What can we use this for? We think that this could potentially revolutionize traumatic brain injury diagnosis and treatment. Because we're designing this to be used in the field, it could enable rapid triage on the site of a car accident, for example. It can help a physician in a hospital monitor response to treatment. So if these biomarker concentrations are not returning to baseline, they can monitor that and switch up their treatment approach. As I said before, it can help to predict outcomes and prognose a patient, so figure out exactly how they're going to be doing 3 months, 6 months, a year down the road. It can lead to precise diagnosis to tell someone exactly how severe their injury is. And it can also lead to accurate enrollment in clinical trials. One of the problems with developing treatments for traumatic brain injury is that enrollment in clinical trials is largely done using the Glasgow Coma Scale. And that scale only divides into mild, moderate, and severe. And it can also be confounded by a couple different factors. So for example, one factor would be intoxication. So if someone is in a car accident because of drunk driving and a physician is assessing that patient using the Glasgow Coma Scale, it's difficult to tell if their impairment is due to a brain injury or intoxication. And so this biomarker device would be able to tell the difference between those two conditions. And so with that, I'd like to thank my lab, especially Dr. Sarah Mena and Dr. Martin DeBeer, and my collaborators, Dr. Fred Corley, who brought this project to us, and Haley Falk, who has been instrumental in helping me test some patient serum samples, which I didn't present about today. I'd also like to thank my funding sources, especially the Precision Health Scholars Award. And with that, I'm happy to take questions.
Great, thank you so much. That was a wonderful presentation. There's one question right now that has come through, and if others have questions, please feel free to type them in the Q&A box. How do you estimate the number of TBI cases that were never reported? Are there post-mortem reports or other sources to make the estimates of the incidence of TBI?
So I'm not really an expert on those kind of methods, I'm going to be honest. So I don't know if I've ever seen like an explicit number for how many TBIs aren't reported. It's just that we know how many are reported, but people say it's an underestimate because it's an underestimate simply because of the way traumatic brain injury presents itself.
Yeah, and I think it's also on a spectrum. So we clearly know when TBI is happening, but then there's other gradients of it and degrees of TBI. So I think this would be a nice way to have an understanding. Let me see. Comment, two very interesting presentations. Thank you. So thank you both to our speakers again. I think the thing that in hearing your presentation, and I've heard it a couple times before and have enjoyed it every single time.
Thank you.
Is I think there's so many different ways where this testing could be implemented, and we've talked about in the middle of a desert, certainly in a war situation, but I also think in a school basketball game or a school football game, and having the ability for the point of care testing. And so I think it's two really great examples of kind of big data, looking through the EHR for identifying patients who are at risk for something before anything happens, and then kind of smaller data point of care testing where we're able to implement and identify who might be at risk for something bad happening or maybe needs some more monitoring. So I think that both are great. And that's more of a comment than a question to you. Are there others questions from the audience as well that are coming through? I know that both of you are, Arun, I don't know if you want to join us as well. I know both of you are looking for the next steps in your career. And so if you've kind of thought like where you would both like to take your science as you're kind of finishing up your programs.
Yeah, so right now we're actually part of the Coulter program housed in Biomedical Engineering. So we're really trying to translate this technology to the clinic. And so we're doing a lot of market analysis and customer discovery right now, and really honing in on the initial context of use of the device. And so that's kind of exciting. As I'm finishing up my PhD, I'm kind of seeing it start to get translated, which is awesome.
Yeah, I can say a word or two about our next steps as well. So yeah, we are trying to collaborate with Intel and try to influence their development of future processors so that they can try to put in certain optimizations which are specific to genomics and specific to sequence analysis. So that's one direction we're looking at. I think Professor Das is also heading a startup which is trying to commercialize some of the work that we have done here. Um, and in the future, I think we are very excited to work with some of our collaborators in the medical school and try to develop better predictors so that we can translate some of our findings to nursing homes and make the lives of elderly patients there better. Yeah. So these are like a few, I think, near-term directions that I'm looking forward to in the next few months.
Is this so where you both are? Because you're both very much on the patient side right now of your research. Is this where you envisioned that you would ever be, or was this something that you kind of came to appreciate at Michigan or as part of Precision Health?
Yeah, I can go first. So yeah, for me, I guess when I started out, I had very little background in biology as well. So most of my introduction to the space has been at Michigan, and I guess through Precision Health, it was a wonderful experience trying to collaborate with several other people working on different areas, and that has really helped uh, like take many of these projects forward and develop very interesting and innovative results. So yeah, I think in the future now, I'm very excited to continue working in the same direction. So trying to solve computing problems in biology broadly. Yeah.
So as a biomedical engineer, I think we tend to be more on the patient side naturally. But I guess what I wasn't necessarily expecting was to be doing like clinical validation and actually translating a device. I always just kind of assumed that I would just be, making a device, and that was kind of as far as I could take it in a PhD. But I'm really excited that I get to translate the device. And moving forward, I actually accepted a position in BD's Technology Leadership Development Program. So I'll be rotating through different locations at BD and different projects, all to do with medical devices in different stages of development. So I've learned more about product development, and I'm very excited about that.
That's very exciting. Congratulations. What has been some of the steepest learning curves that you've had as part of your project, or things that surprised you? I know I didn't have you prepare for these questions either.
Yeah, maybe I can give it a shot. So for us, I guess when we were initially working on this, there was a lot of resistance to change the algorithms from practitioners. And I think what we have come to realize is that there's a very limited space of optimization, like for as a computer scientist, where we have to make sure the existing tools are working as is, and there's little change to what people are already doing. Otherwise, there is a lot of barrier and a lot of reluctance in making use of it. So I think that is, we figured this out luckily early on, and that has driven all our research. So we have not tried to change what people are already using so they can use their own tools as they were before, but take advantage of what we have done. So
kind of
yeah.
the human factor associated with the
Mhm.
processes.
Yeah.
Yeah, I think for me, I wasn't expecting with this device to there kind of be like a chicken and the egg problem. So like measuring traumatic brain injury biomarkers is at the bleeding edge of clinical care for traumatic brain injury. And so I've found that it's difficult, like when you're trying to translate a device like this, it's difficult to justify that this device is necessary because there's not a consensus clinically yet about how these biomarkers are going to be used. But you can't really have a consensus clinically without a device like this or similar, because you need to actually be measuring these biomarkers in the way that you would be in the clinic. And so it becomes this cycle of, okay, well, we can't really get these answers without this device, but you don't want the device without these answers. So that's been something that has been challenging.
Wow. Yeah, that's part of the conundrum with changing our processes. So going back to kind of what Arun said, but optimizing things so that we can advance patient care. And so once again, I want to thank both of you for your excellent presentations. I also want to make sure to thank Tina Krazier, who started and set up this entire webinar and was kind of our person in the background, making sure everything went off without a hitch. We will be sending you a link, everyone that's part of that's listened today, to the recorded webinar in a few days, and then feel free to share those who couldn't make it today that were interested or that you think might be interested in these presentations. And I want to thank everyone for joining us today. And with that, I will have you go and enjoy the rest of your afternoon. It is going to be a warm day out today, but we have cooler days ahead of us. So thank you all for attending.